Welcome to Lecture 9 of Biology 104, where we will discuss plant diversity in the seed plants. In prehistoric times, the seedless vascular plants dominated the landscape. However, in modern-day landscape, we now see the diversity in seeded plants. Seeds are considered to be embryos that are equipped with nutrients as well as a protective coat. Seeds are a very important stage as they allow for dispersal and also for enhanced survival rates in very harsh conditions. Seeds are a key adaptation that allowed for plants to dominate the terrestrial landscape. There are many derived traits that seed plants have in common. These include having reduced gametophytes that develop and remain protected within the sporophytes. These are present in cones if we are discussing gymnosperms or flowers in angiosperms. Seed plants also exhibit heterospory. This means that they produce both mega and micro spores, representative of the female and male spores respectively. Seed plants also have ovules and pollen that protect the female and male gametophytes, and seeded plants also have the development of seeds. These seeds result from pollen fertilizing an ovule. We're now going to take a look at these derived traits. The first derived trait we will discuss is the presence of reduced gametophytes. Gametophytes in general have reduced over evolutionary time and are now considered to be microscopic in most of the seed plants. This small size allows them to be retained within the sporangia. This means that they can remain fully protected from any environmental stress. This also means that they need to obtain their nutrients from the parental sporophyte. In general, having reduced gametophytes allows for an increase in survival ability of these stages. In gymnosperms, the gametophytes develop within the cones, while in angiosperms, the gametophytes develop within the flowers themselves. This slide here gives an overview of the difference between the gametophyte and sporophyte stages in the different types of plants. If you recall, in mosses and other non-vascular plants, the gametophyte was the dominant stage, while the sporophyte was very reduced and remained on top of the female gametophyte and was dependent on the female gametophyte for nutrition. In ferns and other seedless vascular plants, remember that the gametophyte started to become reduced and the sporophyte was the main stage. In seed plants, the gametophyte is completely reduced, usually microscopic, and the sporophyte still remains to be the dominant stage. However, in seeded plants, the gametophytes are going to be protected within either the flower or within the cones. Note that in gymnosperms, we can have ovulate cones, which contain the female gametophytes, and we can have pollen cones, which contain the male gametophytes. We're now going to discuss the next derived trait of heterospory. If you recall from the previous lectures, bryophytes in seedless plants are considered to be homosporous. This means that they only produce one type of spore, and that spore produces a bisexual gametophyte. That bisexual gametophyte can produce both male and female gametes. However, seed plants are considered to be heterosporous. This means that they have megasporophylls as well as microsporophylls. The megasporophylls contain megasporangia that produce megaspores. These will eventually result in the female gametophytes. It's also important to remember that due to the size of the megaspores, each megasporangia produces only one megaspore. With respect to the microsporophylls, these contain microsporangia that then produce the microspores. These ultimately result in the male gametophytes. In this instance, due to the small size of the male gametophytes, one sporangia can produce many microspores. This image here is taken from a research article from Wang et al. 2017, showing the difference between the megasporangia and microsporangia. The megasporangia is labeled as ME in the images, and the microsporangia is labeled as MI. Note in image A the presence of a megasporangia in the center of the image. Also note the presence of a megaspore labeled Mg present within the megasporangia. Also note that there are microsporangia labeled Mi to the right and to the left of the megasporangia. We're now going to discuss the next derived trait, the presence of ovules or eggs. Remember that seed plants retain the megasporangium within the parent sporophyte. The parent sporophyte will provide nutrients, and in this case it also provides an important layer of integument that serves to protect the megasporangium. Gymnosperms will have one layer of integument while angiosperms will have two. The ovule itself contains three main parts: the megasporangium that contains the megaspore and also the integument. Note in the image below the difference between an unfertilized and fertilized ovule. In the unfertilized ovule, you have the megaspore in the center surrounded by the spore wall. This is embedded in the megasporangium, which is then surrounded by the integument. Note the presence of a micropyle, which serves as the attachment point for a male gametophyte. When a pollen grain enters into the micropyle, the male gametophyte will then be released. A pollen tube will then form, and this will allow for the sperm to reach the egg. The next derived trait is the presence of pollen or sperm. The microspore develops into a pollen grain. This results from the male gametophyte being enclosed in a pollen wall. Thus, within each pollen grain, you have a male gametophyte that lies within the center. This male gametophyte will be protected by the pollen wall, which is comprised of sporopellenin. Pollination itself is a process that refers to the transfer of pollen to the ovule. If a pollen grain successfully germinates, this will develop into a pollen tube. This will help to release the sperm into the female gametophyte, and this occurs within the ovule itself. Note in the image below we have the presence of the male gametophyte as well as an established pollen tube. The sperm will then travel into the pollen tube to eventually reach the egg. In this instance, we have a nucleus from both the egg and sperm prior to fusion. This is all occurring within the megasporangium. If you recall, bryophytes and seedless vascular plants had flagellated sperm. This sperm needed to swim within an aqueous medium such as water in order to successfully reach the egg. As a result, many of these plants live in moist as well as semi-aquatic habitats. Seed plants, however, produce sperm that do not need to be flagellated. This is because they can easily travel through the short pollen tube. As a result, most seed plants produce non-flagellated sperm and rely heavily on the pollen form as a mode for dispersal for the plant. However, some gymnosperms do retain the presence of flagellated sperm. We're now going to discuss the last derived trait, the presence of seeds. The seed itself includes the embryo, a food supply, as well as a protective integument. Seeds provide a distinct advantage over spores as a reproductive strategy. Seeds are considered to be multicellular, thus giving rise to a complex plant. Seeds can remain dormant for years, weeks, or months. They have a constant food supply, which increases their rate of survival, and seeds can also travel long distances, increasing the dispersal of the plant. Modern-day seed plants are represented by two clades, the gymnosperms and angiosperms. Gymnosperms have seeds that are exposed on sporophylls that eventually form cones, both ovulate as well as pollen cones. These include most conifer plants. Angiosperms have seeds that develop into mature ovaries or fruit. These are represented by most flowering plants. We're now going to discuss the gymnosperms. If you recall, seed adaptations are also correlated to the following traits: the miniaturization of gametophytes, the seed stage being resistant as well as dispersive, and pollen is also dispersive, which helps bring male and female gametes together. Pine trees themselves are representatives of the sporophyte stage. Remember, these are also considered to be heterosporous, which means that they will produce macro as well as microsporangia. In gymnosperms, the gametophytes develop within the cones. Macrosporangia are present in large ovulate cones, which are depicted in brown in the image to the left, while the microsporangia are present on smaller pollen cones. These are depicted in beige in the diagram. Pollen cones derive from modified leaves called microsporophylls. These leaves eventually bear the microsporangia. Within the microsporangia are specialized cells called microsporocytes. These cells will undergo meiosis to produce the microspores. The microspores are considered to be haploid, thus are represented by the end stage. Each microspore develops into a single grain of pollen. You will have many microspores present within one microsporangia. Ovulate cones, on the other hand, form from modified stem as well as modified leaf tissue. In this instance, the modified leaves are originally called megasporophylls. These will eventually bear the megasporangia. Within the megasporangia, cells called megasporocytes will undergo meiosis to eventually produce the megaspores, also considered to be haploid. These will eventually develop into the female gametophytes that will be held within the megasporangia. Once a cone appears, and if an ovule is successfully fertilized, it can take almost 3 years to produce a fully mature seed. Once the seed is mature, the scales within the ovulate cone will separate, raise, and allow for the seed to disperse mainly through wind. The image to the left depicts the difference between an ovulate or seed cone and pollen cone. The image to the right depicts the difference in the ages of the cones. Note that at the tip of the branch we have a 1-year-old cone, which is very small. Note that we have the 2-year-old cone in the middle, which has the scales folded up to protect the developing seed, while the 3-year-old mature cone will now have the scales raised, which will allow for the seed to disperse. This image depicts the typical life cycle of a gymnosperm. Note that most of the life cycle exists in the diploid or 2N stage, with only a small portion being haploid. We begin the cycle with a mature sporophyte, which is in essence the tree itself. Within each tree you will have branches that will have ovulate and pollen cones. Within the pollen cone you will have the presence of many microsporangia. Within the microsporangia you will have many microsporocytes that will undergo meiosis to produce microspores. These will eventually produce pollen grains. Within the ovulate cone you will have the presence of the ovules. Within each ovule you will have a megasporocyte housed within the megasporangium. This will then undergo meiosis to produce the megaspores that will eventually develop into the female gametophyte. The egg will then be produced within the archegonium of the female gametophyte. If successful germination occurs, a pollen tube will form from the germinating pollen grain. This will allow for the sperm to travel to the egg to allow for fertilization. If fertilization occurs, an embryo or a new sporophyte will develop. Within the seed, you will have food reserves that will help nourish the developing embryo, as well as an integument that will help to protect it. Once the seed matures, it will then disperse. If it is successful in dispersing and reaches the soil, it will then germinate itself to form a seedling plant. This will eventually develop into the mature sporophyte so the cycle can begin again. If we consider the evolution of gymnosperms, we note that within the fossil record there are some seed plant characteristics that appear in various types of plants within the fossil record approximately 380 million years ago in the late Devonian. Archaeopteris is a heterosporous tree, however, it did not have the ability to yet produce seeds. The first evidence of seed-producing plants appear in the fossil record approximately 360 million years ago. Alkinsia is a plant that appears within the fossil record that precedes the first gymnosperms by about 55 million years and the first angiosperms by approximately 200 million years. Note the presence of an ovule in this Alkinsia fossil. During prehistoric time, the Carboniferous period was very moist and was dominated by plants such as the bryophytes, lycophytes, and other seedless vascular plants. This climate became much drier during the Permian era, which led to the success of gymnosperms. This was due to the presence of a thick waxy cuticle that prevented desiccation, the development of seeds, as well as the ability to produce pollen as a dispersal mechanism. In addition, many of these gymnosperms formed important interactions with animals such as insect pollinators. This also helped increase the genetic diversity as well as increase the dispersal ability of the pollen. Gymnosperms in general dominated the landscape well into the Mesozoic era. However, angiosperms began to appear during this time and also began to dominate certain ecosystems, leading to the landscape that we see today.